Okay, so these last few questions here are not nearly as involved. They really just get us thinking a little bit more about some of the material that we went ahead and talked about in these two lectures. This first question here, though, or I guess second official question of the challenge questions in general, is a bridging question between units, which is going to become more and more important now as we enter Unit 3. So I'll remind you that there is a cumulative final in this class, and so we need to start bridging our concepts from other past units to the units here, just so we have that comprehensive understanding of the entirety of genetics. So this question says that exposure to radiation induces mutations in all DNA. I think we all kind of know that from just general life. To determine whether human exposure to radiation results in an increase in recessive lethal mutations. So in order to see if these are occurring in the germline, so in the gametes, scientists can examine the gender ratio of children born to parents exposed to higher than normal radiation levels. Why might that be? So really what this is saying is if scientists want to know whether or not individual germline cells are accumulating recessive lethal mutations because of exposure, they can look at the number of boys born to these exposed people versus the number of girls. That's the gender ratio. And you're asked to try to figure out why that is. Well, there's one type of recessive lethal mutation. So you might imagine a case where we have healthy individuals, and then we have individuals with the recessive lethal mutation. Now, these individuals will survive. And then if we have individuals with a homozygosity for the recessive lethal mutation, these individuals die. There's nothing there that suggests a gender difference. So we have to think a little bit harder here and ask ourselves, well, what types of traits in general result in gender differences? Where have we seen that before? And if a little bell goes off from Unit 1, we begin to realize that we saw gender differences in sex-linked traits. In sex-linked traits, in order for males to show the trait, they only need one allele on the X chromosome because they only have one X chromosome. In females, especially for recessive traits, for a female to show a recessive sex-linked trait, such as colorblindness, she needs to have two recessive alleles. So that's our ticket into this question. Once again, let's imagine that we are interested in recessive lethal mutations. But if we imagine that those recessive lethal mutations are sex-linked, in other words, the phenotype of this recessive allele is death, then what we see is that every male who has this allele, period, every male who has this allele dies because it's recessive lethal. However, not every female who has this recessive allele dies. Why? Well, because there are two types of females that can have this recessive allele. There are our heterozygotes, and they are our homozygotes recessive. Now, for sure, these females die, but these females survive. So since we can have daughters born who have a recessive allele but survive, but we cannot have any sons born who have a recessive allele and survive, in this case, the number of girls born in the next generation will be significantly higher than the number of boys born, giving us a change in the sex ratio. So that's the answer to this question. Recessive alleles that are lethal and sex-linked will result in fewer sons born in the next generation than daughters, and this difference in gender ratio alone can determine that there have been mutations accumulated in the gametes that are recessive and lethal. The last part of this question asks you to think about how such a mutation, radiation-induced mutation, a single base substitution, might lead to no protein being made from a gene at all, to render a protein completely unmakeable. So to do that, we want to start with a sequence of DNA or messenger RNA to kind of wrap our mind around this. And here's a very, very brief sequence of an extremely small protein, just for the sake of example. And we're going to go through the same exercise we did for the first challenge question. We're going to first divide this into codons, establish our reading frame, and then we're going to translate this. The ATG start codon we know as methionine. That's followed by a TGG or UGG in the messenger RNA. And TGG codes for tryptophan. And then CCG codes for proline. GAC codes for asparagine. And then TAA or UAA is a stop codon, so that's the end of the sequence. And the reason why I'm pausing there between each amino acid that I name is because I have a textbook open right in front of me with genetic code, and I'm looking up what these codons are. So remember, there's no expectation that you know these codons. You'll be provided with a genetic code on any exam that you get from me. So this is what the protein should normally be. Now I want you to imagine that we have a single transition at nucleotide number 6. So nucleotide number 6 is right here. And if it's a transition, that's all I need to tell you. I don't need to give you a nucleotide because transitions remain in class. And so if we have a guanine, which is a purine, we're going to mutate to the adenine, which is another purine. So we mutate that TGG into a TGA. That's a transition at nucleotide number 6. Now I'll go back to my genetic code and look up what TGA codes for. And TGA actually codes for a stop codon. And so now, essentially, this protein, quote unquote, is actually a methionine only. And methionine only is not a protein. Methionine only is an amino acid. And so this is an example of how a single base substitution induced by something like radiation exposure could lead to no protein being made at all. Yes, this is a nonsense mutation, but it's a nonsense mutation in the second amino acid, meaning we don't make a protein. We don't make a chain of amino acids. We call for methionine, and then we're done. That's going to truncate this protein completely from that mutation forward, and no protein will be made from this mutant. We'll see other ways of causing mutations like this that rely more on gene regulation than gene expression next week. But for this week's material, this is the best example of how a single base substitution can result in no protein expression whatsoever. And finally, to wrap us up on these challenge questions, question 3 says, in the lecture, I say that information almost never flows backwards from RNA to DNA. Why do I say almost never? Can you think of any natural process where DNA does indeed flow backwards from RNA to DNA? Again, I'm stressing here the comprehensiveness of the course and the cumulative final, asking you to bridge concepts from Unit 2 here to Unit 3. Now, I'll admit this isn't too much of a bridge because this was the very last set of concepts that we covered in Unit 2, and we're in the very beginning of Unit 3 here, so we're just bridging from right before the exam to right now. But hopefully you do remember what we talked about with regards to viruses and retroviruses specifically. Retroviruses such as HIV rely on a process called reverse transcription. And in fact, they carry around with them their own enzymes, their own reverse transcriptase enzymes. And reverse transcription takes the RNA genome of the virus and reverse transcribes it into a double-stranded DNA copy of that same viral genome. It's that DNA copy that can then insert into the host's genome and lie dormant in a lysogenic-like state until the infection emerges as full-blown AIDS and causes symptoms for the patient. So yes, in the natural world, we do see genetic information move backwards from RNA to DNA in retroviruses, but that's the only known example where such a thing happens. In all other living systems, it's a unidirectional flow from DNA to RNA. And finally, for number 4, if I must suffer an indel mutation in a gene, specifically an insertion of a base, I really hope that I have an insertion in one place and a deletion somewhere else, or the other way around, a deletion in one place and an insertion somewhere else, really, really close. I don't care so much which base is deleted or which base is added. I more care that they're close together. Why? And so in order to address this question, we once again need to have a sequence in front of us that we can use as a model. So here's a sequence that I just threw together. It definitely starts with a start codon, and it intentionally ends with a stop codon. And we can take the intervening bases in groups of 3, or our triplets or our codons, and that's our reading frame. And we could translate this with a genetic code. I'm not going to take the time to do that now, but certainly you can use this as practice and decode this into its amino acid sequence. But here in this question, what I'm saying is if I have to have an indel mutation, let's say that I have a deletion, let's put it here. Now we can see that there's a pretty drastic effect on the reading frame there. Let's reestablish the reading frame. There's the start codon, there's the second group of 3, there's the third codon, the fourth codon, the fifth codon, the sixth. We're blowing right by the stop codon, and we just keep on going. There's our frameshift mutation. And so what I'm saying in this question is, if that has to happen, remember that scrambles the protein sequence from the point of the deletion all the way down the amino acid chain. Every single amino acid is going to be radically different. This protein is going to be radically changed and non-functional as a result of this mutation. And what I'm saying is, what I would like to happen, if this is necessary, is if you're going to delete something somewhere, please, 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 let me have another insertion somewhere close. So maybe like right there we would put a thymine. Please let that happen. So let's see what the effect of that insertion would be now. So there's our first codon, the start codon. There's our second codon. We're still suffering the effects of the frameshift. You can see that after the frameshift this was a GTC, and even after my second mutation that I've begged for, it's still a GTC. So the frameshift is there. But now here's the third codon. It's a TAT codon because of the insertion. And after that T, look what happens. We revert right back to the same frame, because we deleted one nucleotide in one place and added a nucleotide elsewhere but someplace close. We shifted the frame and then shifted it back to its original frame. So, to be fair, is this a mutant codon? Absolutely. But in the first deletion, the entire chain of amino acids was changed from that point of deletion on. However, when we get the second, what's called a suppressor mutation, then we reestablish the coding region and we go back to our original frame. All of these amino acids are proper, and really we're only suffering the effects of a single, essentially missense mutation, where one amino acid has been substituted with another. So frame shifts get quite abstract in this way, in that they can scramble entire amino acid sequences. But if you shift forward and then shift back in a close region, you can limit the effects of the frame shift, and that's exactly what we're getting at here. So hopefully you did try these on your own first, and hopefully these explanations were clear for you. I look forward to getting back in town very, very soon and meeting with you all for a full and normal week next week, where we talk about gene regulation, one of my favorite topics in all of molecular genetics, and quantitative genetics as well. Until then, thanks for watching.